Hello, I am Rob David from Thermo Fisher Scientific, and this is Double Helix Diaries. In this episode, meet Dr. Archana Gupta, an esteemed scientist with experience in infectious disease. Today, Archana will explore with us the capabilities of multiplexed PCR. Well, Archana, welcome today to Double Helix Diaries. We are so happy to have you here.
I'm very excited to be here. Thank you.
Excellent, excellent. Well, let's dive in, shall we? Most have heard about Sanger sequencing, but many, I think, are unaware, as you and I are, that there's a lesser known application for capillary electrophoresis called fragment analysis. If you could, please tell us a bit about fragment analysis and its application.
So you're absolutely right. Fragment analysis is a lesser known application of capillary electrophoresis, but an equally powerful one. And in its most simplistic way, if I had to describe it, it involves analysis and resolution of DNA fragments by their size. So in terms of the workflow, you start with isolated DNA, then you PCR amplify it using fluorescently labeled primers. Now, the amplicons that you get that are fluorescently labeled, then you mix it with your size standard that is also fluorescently labeled but in a different color, and then you resolve it on a CE instrument under high voltage. And so what that allows you to do is to look at these different size DNA fragments that you have PCR amplified with as little as a resolution of 5 base pairs. And so the multiplexing capabilities that you get with this combination is incredibly powerful.
I don't think I've ever heard someone elucidate it quite like that. And I've been doing it for years, so thank you. Thank you for that. I understand that you have specialized in multiplex PCR as well. So how is multiplex PCR used or identified with fragment analysis?
So with multiplex PCR, so PCR essentially is an upstream step of fragment analysis. You can do multiplex PCR not just by multiplexing different targets that you want to look at in a particular DNA fragment, but you can also multiplex at the sample level. So essentially, you can multiplex around 400 targets for one particular sample, and then you can multiplex up to 4 samples within the same well. So essentially, you're looking at 4 times 400 amplicons that can be resolved using multiplex PCR and fragment analysis, which is absolutely incredible. And I am not aware of any other technology that allows you to look at DNA fragment or DNA size by this method.
Wow, you're absolutely right. First time, and I admit my background was not in molecular biology. I was a chemist by trade. And so when I first was introduced to PCR alone, that was amazing. But then the idea that you could add essentially thousands of those same reactions in the same well and then be able to split them out and detect them with fragment analysis was truly amazing.
I will add over here that you do need to optimize for the PCR reaction itself, right? Because if you're using that many primers, you need to make sure that they don't cross-react and that you don't get aberrant, amplicons. So with that little, caveat that there is involvement of optimization of the PCR reaction, but if you do nail it down, you can go up to thousands of essentially amplicons where you're multiplexing at the target as well as the sample level within a single well, when you do capillary electrophoresis and multiplex PCR.
Yeah, but optimization, I mean, that's what makes it fun. That's why we're in science, right?
Yep.
So more optimization, the better. We get to play around with the reactions. Now, building more on this, on multiplex PCR, what are some of the benefits and advantages of utilizing multiplex PCR in comparison to conventional PCR approach?
Yeah, so as I mentioned, the power of multiplex PCR with CE really lies in the order at which you can multiplex. So you can easily go I mean, technically or theoretically, you can resolve as little as 1 base pair fragments apart, but comfortably about 5 base pairs apart. So if you're looking at fragment sizes that are five base pair apart, you can go up to 400 targets, like I said, and then you can multiplex at a sample level up to 4 different samples. And that's something that you would never be able to do if you did conventional PCR, or for that matter, even if you did qPCR. There is no way that you can get that order of multiplexing with any other technology for looking at different DNA fragment sizes.
So I understand your background is in infectious disease. So aside from what I'm assuming you have used it for before, which is to identify pathogens, what other potential uses are there for fragment analysis?
Yeah, and I'm glad you asked that question, because really the applications are limited by the imagination of the researcher. My interest, of course, is in pathogen detection, and we had actually developed an app note around this to show that you can detect various respiratory pathogens, several tens of them, using fragment analysis. But you could essentially use it for anything. One of the popular applications is STR analysis for cell line authentication. So for example, think about cell therapy now, right? Where you isolate cells from a patient, and then before you want to put it back, you want to make sure that it is in fact, the cells of the person that it needs to go back to. And so you can do STR analysis using fragment analysis, and then verify and confirm that the cells belong to the donor that you had isolated the cells, the sample from. You can also use it for microbiome analysis, so detecting various bacterial species. That's a really hot topic right now, because it's known that the microbiome, the way the microbiome is in an individual can influence their immune system. And so it becomes a really interesting application if you want to look at what kind of different species exist in a person's gut. Gut microbiome, for example. The other thing that you could do is do variant analysis to determine pharmacological response to various drugs. Or essentially, if you want to look at different mutations that cause a particular disease, you can pick out those particular, hotspots in the DNA, and then design, primers to that and see whether, that particular region amplifies, and therefore, yes, that mutation exists or does not. So really, like I said, there are diverse applications, and you just really need to think about the question that you need to answer.
Wow. So pharmacogenomics, pathogen detection, gut microbiome. I mean, it really, as you said, it's really just limited to the imagination. I love it. Well, given we're scientists here, as you might imagine, we do our research. And I heard that you are rather adept at describing what we do here at Thermo Fisher with our budding would-be scientists.
Yes, I'm a very passionate scientist, and I have a toddler. He just turned 5 a couple of months back, but I really love sharing my love for. I really love sharing, my love for science, because I think there is wonder all around us. And I remember when I was a little child, my interest in science really came from, looking at the touch me not plant, Mimosa pudica, where you would touch the plant, the leaves, and it would close up, and then it would open up again. And so I think from a very young age, if you really start observing the world around you and you break down concepts, it can spark interest and make you, because I think science or research is imaginative. And so if you can break down concepts for little kids, I think you can get them to be interested in science and research from a very young age, and they might give you ideas about your research that you couldn't have thought of yourself. So one of the examples that I guess I can share over here is that I was trying to teach my 5-year-old son about what DNA is. And so I was telling him that just like he reads books, and the letters in the books are from A to Z, DNA is the book of our life, and it's coded in 4 letters, A, T, G, and C. And many a times, diseases arise when things go wrong in the way these letters are sequenced, right? So instead of, say, A, T, G, if the letter becomes A, C, G, or if there is I tried to explain to him what a mutation is, but it was essentially my attempt in trying to build for him the concept of why is it that we all look so different, but if him being my son has my eyes, and the lips of my husband, what is it that he carries from myself and my husband that makes him look similar yet so unique? And so, it's a combination of these two books, my husband's DNA and my DNA, that makes him have his own unique DNA with his own unique, letters. So yeah, I try to do my best in breaking down these complex, concepts of science for kids, and I'm still, work in progress, but I think I find it incredibly fascinating and creatively challenging as an exercise for myself.
Wow, that makes me want to be a kid again, so I could learn from you. Also, I should send my daughter over to your house so she could hear those stories as well.
I do teach. I go and volunteer at my son's school to teach pre-K kids on science, and I absolutely love it because they're just so, starry-eyed and curious, and I think they inspire me, to want to keep doing this more and more.
You're right. You're absolutely right. It's the best feeling when you're teaching kids and you see that light bulb go off.
Yeah.
Oh, man. Well, Archana, thank you so much for joining us today. I've thoroughly enjoyed our conversation.
Thank you for having me.